Good afternoon. My name is Giulio Caravagna and I'm here to present my work on subclonal reconstruction of tumors by using machine learning and population genetics. This is a work that is being carried out while I was a postdoc at ICR London before moving to the University of Trieste and starting my own lab on cancer data science. In the context of cancer evolution studies, what we usually care about is trying to use sequencing data to understand the complexity of the cancers, how many populations are present in our sample, what is the make the difference, why do they behave differently under therapy, why some of them probably lead to metastasis while others don't, etc., etc., etc. So understanding what we call the complexity of intratumor heterogeneity through the lens of evolution. Characterizing the clonal architecture of the cancer by accessing somatic variant data and understanding fundamental questions about how tumors grow, progress, and respond to therapy. In the particular context of this presentation, I'm gonna discuss the problem of determining how many special subpopulations are growing inside a single biopsy from whole genome sequencing data. The classical subclonal deconvolution problem is very famous and has been usually defined by thinking about variant allelic frequency, which is the ratio of alleles that I can access through sequencing that harbor a particular somatic variant relative to all the alleles that I can map on top of a certain variant. This is therefore a fraction and can be modeled very clearly by some binomial observational model in which we condition upon the depth of sequencing and the variant allelic frequency. So if you think about the problem of understanding how many populations are in a cancer, if we imagine that we are working with populations that are present in 50, 30, and 20% proportions, we can think that our variant allelic frequency across all the cells that I can sequence from this population can be expressed as a mixture model where I have some mixing proportions and it's sum up to 1, and components which are associated to these binomial likelihoods. Therefore, I'm actually clustering the variant allelic frequency spectrum. Each one of the colors should give me a mixture distribution. The peaks of this mixture distribution, the binomial parameters, would give me access to the proportion of cells with all these mutations, and therefore on the clonal architecture of the sample. The truth is that the model is a little bit more complicated because in general one needs to think about tumor aneuploidy, which is the relation between copy number states and variant allelic frequencies, and bulk sample purity, so the fact that in a bulk experiment we also sequence normal cells. But for the sake of understanding the point of this paper, this is completely irrelevant. So while we can think from a machine learning point of view that a generative model based on binomial mixture is perfectly fine, the truth is that we're completely neglecting the fact that certain things are known about tumor growth. Tumors start from a single cell in a process of cell divisions where more mutations are accrued every time a daughter cell is born from the parent cell. Therefore, if we think about annotating somatic mutations on top of a cell division tree and sequencing and therefore drawing the site frequency spectrum, so simulating the variant allelic frequency for the cells that we are likely to sequence, the 4 cells at the end of the cell division process, well, we see that there is a clear distribution with a peak of clonal mutations and subclonal peak of mutations that follow certain characteristic distributions. Mathematically speaking, these distributions can be studied performing a branching process model over the stochastic Uriel-Delbrück model. In this case, we can find that the probability of a certain set of variants to be a frequency m depends on a distribution which is called the Landau distribution, whose approximation follows the 1 over f square tail type of distribution. So if we think about every clonal expansions in the tumor, there will always be a neutral tail inside each one of the clonal expansions that characterize the tumor, because those are just a byproduct of the clonal evolution model. Therefore, this part of signal will be in the data, as we can see through simulation of whole genome sequencing assays at the coverage of 120x. In our work, we have taken together these ideas and we have implemented the mixture model in which we have beta distributions that are used to model continuous values for variant allelic frequencies, together with power law tails as predicted by population genetics. Therefore, our model can capture a tumor with a set of clonal mutations, like clone 1 in the figure here, and neutral mutations, like a Pareto power law tail here, and ongoing subclonal expansions, like clone 2 here. This is what we call a subclone in the context of our model-based tumor subclonal convolution framework. Technically, their model is fit by a variation to a maximum likelihood, which uses moment matching for beta distributions, and confidence is available through bootstrap procedures. Parameters of the model fit can actually be used to learn the parameters of a stochastic branching process model of tumor growth, which are tumor mutation rate, selection coefficient of the subclones, etc., etc., etc. The tool is available as an open source R package, which you should download and test with all the data that we release together with the tool. We have used this method to analyze a number of samples available in the field, new data, as well as large cohorts data. What we have found is that results considered to be extremely solid are actually improved by our analysis. In the context, for instance, of the sample on the left, which is a breast cancer whole genome 200x, more or less, we find that one clusters identified by our method associated to a neutral tail, like the gray cluster there, is essentially the same cluster that in the original analysis is phased at the level of somatic mutations to be part, evolutionary speaking, of all the other clusters found in the data. This is the classical characteristic of a polyphyletic lineage, which is the definition of a neutral tail. This, we think, validates our inference and the fact that that cluster is not a third subclone in the sample, but is actually the neutral tail of one of the three clones in this plot. We have in general found also that if we look at large-scale whole genome sequence data like the one available in the PICO cohort, what we actually find is that if we take the highest quality samples in terms of tumor purity and median coverage, we do find neutral tails in the data, which means that the deconvolution should really think about this type of signal in the data before assessing the actual number of subclones in the data. To conclude, we have shown that population genetic models that include cellular growth and give expected VAF distribution for neutral mutations, they improve over generative methods that are just data-driven and in general simplify the result of the analysis. In the paper we discuss the relation of this analysis to purity and coverage, spatial sampling for multi-region sequencing, comparison with other approaches, and a number of real data examples. Follow up our lab website for support for MOBSTER. Thank you.